Okay, today's unit we are going to discuss blood. So blood is going to be considered one of our connective tissues. If you remember from the histology unit in Anatomy 141, then you'll remember discussing your different types of connective tissues. So being a connective tissue, it's going to be very important within the body. It's responsible for transporting things such as nutrients and food. It will transport oxygen. It transports electrolytes, cellular waste products, and hormones, and also will transport carbon dioxide back to the lungs to be expired. It's going to circulate disease-fighting chemicals that are going to be known as antibodies and cells that are going to help fight these different diseases and things within the body, and those are going to be your white blood cells. It's also going to help you maintain your homeostasis. So it'll pick up on things like acid-base balances. So if it picks up that your blood's becoming too acidic, it will increase your respiration and also work with the urinary system in order to get rid of things such as carbon dioxide and waste products in order to bring the base or the acidic blood back up to more of a neutral homeostasis and back to where it needs to be. It also circulates cell fragments, which are going to be your platelets. These are going to help with you stopping any type of hemorrhaging within the inside of the body or the outside of the body, so it stops bleeding. It also distributes heat throughout the body, and it's going to help maintain, like we said, that proper pH. Now here, the first thing, if you're going to the doctor and they're taking blood, what they'll do is they will draw that blood. First, they're going to put a tourniquet around your arm. After they put the tourniquet around your arm, then they're going to look for a vein to pop up. So they're going to draw the blood with a syringe from the vein, and they're going to put it into a test tube. Inside that test tube, they're then going to seal it and put it in the centrifuge where they spin it. Once they spin it, it's going to use gravity to cause the heavier substances to fall to the bottom and separating it from the lighter substances. So when it separates, it's going to look like the diagram that's to the right. And so you'll see that the top portion is going to be made up of plasma. Now, plasma is going to be made in the red bone marrow. That's also where you're going to have your red blood cells, white blood cells, and platelets made. So the plasma is going to be about 55% of your whole blood. It's going to be the least dense component, and that's why you're going to find it towards the top. The buffy coat is going to be the coat or the section that you see in between the more yellowy color on top and the red erythrocytes that are on the bottom. The buffy coat is going to be made up of the white blood cells, so your leukocytes and some of your platelets. And this is going to be about a little less than 1% of the whole blood. The bottom section is the erythrocytes. These are going to be 45% of that whole blood, or what's considered to be your hematocrit, and that's going to be the most dense part. Now, when you draw blood, or if you get cut or hurt and you look at the blood, it looks like it is a homogeneous mixture. But actually, it can be separated out according to the heavier substances, and so that's where you see the red blood cells sinking to the bottom. Now, if you were to take some of your blood and put it on a slide and look at it underneath the microscope, you would also be able to see these red blood cells on that microscope slide and be able to tell that the blood or the liquid isn't all red, it's just actually the red blood cells that are giving it this color. All right, so with the leukocytes, the erythrocytes, and the actual platelets, all those put together are going to be what make up your formed elements. Now, with the composition of blood, you have, of course, your red blood cells, the white blood cells, and platelets like we just saw, and then there's going to be the liquid portion. That liquid portion is going to be the plasma. And of course, you can see it separated here. Typically, 45% of that is going to be actual cells, and that's also referred to as your hematocrit. So when they look at the hematocrit, they're looking at the different types of cells there. And then 55% of the total blood sample is going to be plasma. So if you look at the slide over to the side here, you'll see different things that you will see in a blood sample when you look under the microscope. You're going to see the red blood cells, and of course, you're going to see platelets. Those are going to be just pieces of broken up red blood cells. And then you'll see white blood cells, and there's going to be several different types of white blood cells, which we'll get to and talk about later. Starting off with the blood cells, all of them start off with what we refer to as the main stem cell, or the mother of all stem cells for blood, and that's going to be the hemocytoblast. So we see the hemocytoblast, it is a pluripotent stem cell. Now, when you go through, if you're asked this question, go with hemocytoblast, that is the proper name. Now, being a stem cell, that means that it can turn into several different types of things. Now, with this hemocytoblast, if it turns into a myeloid stem cell, from there it could differentiate, and it could, when it's exposed to the CFU, it could become a pro-erythrocyte. With a pro-erythrocyte, it's going to eject its nucleus as it matures and become a reticulocyte. That reticulocyte continues to mature after it's ejected its nucleus and becomes your red blood cell or your erythrocyte. Now, lots of times you might see red blood cells abbreviated with RBC. That simply just stands for red blood cell anytime that you see it. Now, if the myeloid stem cell becomes exposed to the CFU factor, then that is going to make it become a megakaryoblast. That megakaryoblast then will form into a megakaryocyte, and the megakaryocyte then will break down into pieces, and that's what becomes your platelets. If it's exposed to the CFU-GM, then with that colony-forming unit is going to either become a eosinophilic myoblast, and then that matures into a eosinophil. It can become a basophilic myoblast, and then that will mature into a basophil. It can become a regular just myoblast and turn into a neutrophil, or it can become a monoblast, and then once it matures, turn into a monocyte. Now, with this myeloid stem cell, sometimes it might go directly into what's known as a mast cell. Mast cells are going to be very important because they're going to help produce histamine when you're having allergic reactions. With going back to the monocyte, with that monocyte, it can also become what's known as a macrophage. And then those macrophages are going to be very important for engulfing certain types of debris and engulfing different types of organisms that the body needs to get rid of. Now, if that hemocytoblast goes the route of a lymphoid stem cell, then it can become either a T lymphocyte, B lymphocyte, or an NK, which is a natural killer. Let's move this up. So with the lymphoid stem cell, if the T lymphoblast then matures, it becomes what we know as a T cell or a T lymphocyte. If the B lymphoblast matures, it becomes a B lymphocyte or what we refer to as a B cell. These are going to be the ones that actually make your antibodies. And then if the natural killer lymphoblast matures, it becomes a natural killer cell. So, talking about these different types of leukocytes, leukocytes is going to mean white blood cell, and you might see that abbreviated with WBC. These are going to be important because their job is to fight diseases. So they'll fight things such as viruses, bacteria, fungi, protists, and even cancer. Those natural killer cells, for example, are very important in fighting cancerous cells that are within your body. And the way they do that is they go around and they'll poke holes in the cell membrane of those cells, which causes them to rupture and destroys them. Okay. Formation is going to be regulated for these white blood cells by certain hormones. So we have what's referred to as interleukins and colony stimulating factors, or CSFs is going to be how they're abbreviated. So these things are actually going to help them form and mature into specific types of white blood cells. You're going to have 5 total types that we're going to discuss that circulate in your blood. And so looking here at this picture, you see connective tissue, you see a capillary, and you see that going through that capillary, it is going to have openings. Those openings are not going to allow the red blood cells to get out. Now, they can diffuse their products across the capillary, but the red blood cell doesn't actually exit through that capillary. What you see here is going to be the white blood cell or leukocyte that's going to be able to actually maneuver its shape and fit its way through the holes that are found in these capillaries in order to go into the tissues and search out whatever it's looking for, whether it be a bacteria or some type of protist or maybe a virus. Now, talking about these different types of cells, the first type that we're going to talk about is going to be a neutrophil, and about 60 to 70% of your white blood cells that are going to be found in a blood sample should be neutrophils. You'll be able to tell these because they have a lobed nucleus, and they're going to be referred to as granulocytes. Granulocytes mean that you can see these granules that are going to be within it. And so it looks very grainy. So you see these cytoplasmic granules, and they often stain purple. Now, if you're looking at a blood sample, it does often look like this, except for you won't be able to see the white blood cell in there. It'll kind of just blend in. That's because the white blood cells actually have to be stained in order for you to see them. So if you were just to cut your finger and, let's say, put some blood on a slide and look underneath the microscope, you would definitely see the red blood cells, but you would not see these leukocytes that are there until you stain them. All right, so with the neutrophil, one of its main jobs is to engulf bacteria. And in order to do this, it's going to use its lysosomes, which are going to be in here, and that's what makes it look really grainy, to actually kill these bacteria once they engulf the bacteria. The next type that we're going to talk about is going to be the eosinophils, and this is going to be about 2 to 4% of the white blood cells that you find in the blood that you take, the whole blood. You definitely see cytoplasmic granules here, so this one's also a granulocyte. These are actually going to stain a reddier color, and notice how it looks like it's full of small little granules or bubbles within inside of it. And then looking at the nucleus, it has this distinct nucleus that some of my students have often referred to as kind of looking like a dumbbell. And so it has this binucleated look or structure to it. This is going to kill certain types of parasites. And the way it does it is it gets close to, let's say, a parasitic worm, and it's going to dump digestive enzymes onto that parasitic worm in order to destroy it. The next type that we see here is going to be a basophil, and this is going to be anywhere from 0.5 to 1% of the white blood cells that we see in a blood sample. These are also going to have cytoplasmic granules, and these are going to stain a deep blue color. So with this type of cell, you know it's a basophil when it looks like it's full of all of these granules, and it's a much darker color. This is going to release things such as heparin. Heparin's going to prevent blood from clotting, and it also helps release histamine, and that's going to increase blood flow to an area in order to get more white blood cells to that area in order to clean up whatever needs to be cleaned up or destroyed. The next type of cell is going to be a monocyte, and this is about 3 to 8% of your white blood cells that you'll find in a blood sample. They have what's referred to as a kidney or kidney bean type nucleus. It's going to be oval or lobed shaped. This type is going to engulf internal parasites and other cellular debris of maybe damaged tissue or old worn out cells that it might come across. The next type is your lymphocyte. This is going to be about 20 to 25% of the white blood cells in a blood sample. They're going to have a large round nucleus. Notice how this nucleus takes up almost the entire size of that cell. And they're going to be slightly larger than the red blood cells that are around it. And these are going to be very important in immunity. And we'll discuss these much more when we get to the immune system. Now, white blood cell counts are going to be something that's very important. You might have a WBC count. If you were to go in and have a blood test taken, let's say if you've been sick and they want to figure out what type of organism is making you sick, they might go in and test your blood for these. So on normal, you're going to have anywhere from 5,000 to 10,000 per microliter. That's going to be found in a unit of blood. Leukocytosis means that you're exceeding the 10,000 per microliter, sorry, I went blank, per microliter of blood. And so that means that you might have some type of active infection at that point. And so they're going to look to see specifically what type of white blood cells that you have a lot of. If it's leukopenia, that means it's going to be below 5,000 per microliter, and that could lead them to, you might have some type of illness or disease that they need to look into because your body's not making enough of those white blood cells. And so here's just a review of the different types of white blood cells and what happens if you might have a high count of these or a low count. So with the neutrophils, if you have a high count of neutrophils, you might have a bacterial infection. Your body might be fighting a burn or some type of stress and inflammation. If you have a low count, it could be that you've had radiation exposure, maybe drug toxicity, a vitamin B12 deficiency. You might have lupus. If you're looking at your lymphocytes and you have a high number, that means that you possibly could have a viral infection. Some leukemias, an infectious mononucleosis, so you might have mono at that point. If you have a low count of lymphocytes, this could mean that you have had a prolonged illness. It is an indicator for HIV. It could be some sort of immunosuppressant, or it could be because you have a treatment with cortisol that you've been taking for some other type of condition. With the monocytes, if you have a high number of monocytes, it could be due to a viral or a fungal infection. It could be due to something such as tuberculosis, some leukemias, and other chronic diseases. If it is low, then that means you might have a bone marrow suppression or treatment with cortisol again. With the eosinophils, these could be found in things like allergic reactions, parasitic infections, and autoimmune diseases if you have high counts. If you have low counts, it could be due to drug toxicity, stress, or acute allergic reactions. And with the basophils, if you have high amounts of these, it means that you could be having an allergic reaction, leukemias, cancers, or hypothyroidism. If you have low counts of basophils, it could be due to a pregnancy, ovulation, stress, or hypothyroidism as well. So going in, red blood cells can also be called the erythrocytes. They are going to be a biconcave disc. That means that they will have a larger section around the edges and that they're going to be concave in through the center. They kind of remind me of a type of float that you would get into, or if you were going to go tubing or ride down the hill in the snow, this would be like those types of tubes. So it does have a thin membrane through the center, but it is not going to be full like on the outer edges. Mature red blood cells do not contain a nucleus. Remember, they eject their nucleus as a reticulocyte, so they will not have a nucleus. If they don't have a nucleus, that means they also don't have DNA. So if they were to take blood samples at a crime scene to use someone's DNA to figure out who did it, it's not going to be coming from the red blood cells. It will be coming from the DNA that's found in the nucleus of the white blood cells that's in that blood. Okay. 1/3 of the cell volume with these cells is going to be a protein called hemoglobin. Hemoglobin is going to be very important because it's able to attach to oxygen molecules and carry it throughout the body and release it into the tissues that need it. Now with a red blood cell, its average lifespan is going to be about 120 days. Once it has lived its lifespan, then it will be cleared from the bloodstream, and some of the red blood cell, as it's destroyed or disassembled, will be reused, and other things will be excreted from the body. And we'll talk about that in a little bit. Okay, so looking here, this is a hemoglobin molecule. It is actually made up of 4 proteins. It's going to have 2 B globin chains, and it's going to have 2 A globin chains. And so these chains will then hook together and form the structure of hemoglobin. You see the heme group, which is going to be that red center portion, and that's going to be the portion that's able to lock onto the oxygen molecule. Now these are going to be formed in the red bone marrow. Red bone marrow, if you remember from Anatomy 141, when we talked about the skeletal system, is going to be found in the epiphysis or epiphyses, which are going to be the ends of the bone. It's going to be found in spongy bone. And so with that spongy bone, the red bone marrow is going to create red blood cells, white blood cells, plasma, and platelets. And so this is going to be where your red blood cells are going to be created. A hormone called erythropoietin is going to control the rate of red blood cell production. It also will help control the rate of all the other things that we just mentioned. Now, lots of times this is going to be abbreviated with EPO. So if you see EPO, that's the erythropoietin, and that's what it's talking about. Now, with this, if you were to get into an accident, if you had a cut, if you lost a lot of blood, the yellow bone marrow that's found in the medullary cavity of the long bone or the shaft part of the bone can actually be converted over into red bone marrow in order to get the plasma and red blood cells and all of those things back up. Now, once the blood has come back to normal levels and you have enough, then that red bone marrow that's in that diaphysis or in the shaft part of the long bone will convert back over to yellow bone marrow, and homeostasis will take over, and then it'll just be found in the spongy bone of the epiphyses. All right, so if your body detects that it has low blood oxygen levels, it's going to send signals to the liver and to the kidney. That, in turn, is going to create the EPO, or the erythropoietin. That goes into the bloodstream, and the bloodstream is going to carry it to the red bone marrow. Now, looking here at this bone that we see, notice the red bone marrow is showing at the end, and we call the end of the bone the epiphysis. That's going to increase the production of red blood cells in that spongy bone with the red bone marrow. Once those numbers increase, oxygen carrying capacity will go up. That signal gets sent to the liver and the kidneys that are producing that EPO, and it shuts down the production, so it does not need to make any more. Okay, and this is just another diagram going over talking about that and showing that. All right, so in order to make a red blood cell, first you start off with the stem cell. Remember that stem cell is going to be called a hemocytoblast. Know that name. Then it's going to turn into a committed cell. Remember, once it's committed to becoming a red blood cell, it's going to be known as an proerythroblast. That proerythroblast starts to develop in its first phase. It goes through ribosome synthesis, where it's going to make specific proteins that are going to be needed, mainly the hemoglobin. With that is called the basophilic erythroblast. It then goes into phase 2. That's where the hemoglobin starts to really accumulate there. And with that, it becomes a polychromatic erythroblast. Then it turns over to an orthochromic erythroblast. And notice how that nucleus is starting to shrink down at this point. Then it's going to eject that nucleus and become a reticulocyte in phase 3. So the reticulocyte is going to be the last phase of maturity before it becomes a full erythrocyte or red blood cell here. And so once it becomes that erythrocyte, it's going to be mature and it's ready to do its job. All right. So what happens when that red blood cell has lived its lifespan? It's 120 days old and it is ready to retire. Well, it's going to be destroyed in the liver and in the spleen by cells that are referred to as macrophages. And so here, we're going to look at this. We have that red blood cell in the diagram. It's first going to be broken down by the macrophages and is put into two different parts or two different substances. You have the heme portion, which is going to be iron plus protein. The iron is going to go to the side. It can be reused. And then that protein is going to be turned into biliverdin. From there, biliverdin then becomes bilirubin, and then the bilirubin goes into bile. Bile then will be stored in the gallbladder and then used to help break down fats in the duodenum, or the small intestine. Looking over here with the protein that is broken down to the globins, that is then going to be reused in the body in order to make more red blood cells. Okay, so iron's reused. And then you have that globin, which is digested. And then, of course, what I meant by reused is it's going to break it down into amino acids. Those amino acids are then going to be able to be reused in other proteins that are being put together at the ribosomes. All right. Looking at this picture here, we see the circulation. It's just going to be another picture that you can see what's going on. And so it circulated for about 120 days. So that red blood cell is going to go through its death phase and phagocytosis. It gets broken down. We have the globin, which then gets digested and goes into amino acids. Those amino acids then will get reused into whatever protein that your body needs to make. The heme is going to get broken down and it turns into either iron, which then can be used again, or biliverdin, which turns into bilirubin. That bilirubin gets secreted into the small intestine from the gallbladder. So with red blood cells, a normal value of red blood cell count is going to be about 5.4 million per microliter in males and 4.8 million per microliter for females. And so males are going to have a higher blood volume than females. With this higher blood volume, it means that it does carry more oxygen. With more oxygen, of course, they're going to be able to have a better, almost what you would refer to as an endurance because of this, because they're carrying more oxygen. Anemia means that you have too few red blood cells, or you have too little hemoglobin, and you might need iron in order to up that hemoglobin in order to increase its affinity for oxygen. Now, B vitamin deficiency could cause this. Vitamin B12 and folic acids are going to be very important in helping to create red blood cells. You could be anemic or have problems because you have been bleeding. Let's say you could have been cut or been in an accident and lost a lot of blood, been hemorrhaging. Or, like I said, you might just simply have an iron deficiency. And so with an iron deficiency, you can take supplements for that, and you can change your diet in order to get the iron count back up. Now, sickle cell disease is going to be a genetic anemia. That means its oxygen carrying capacity of the blood is going to be reduced. Some of the red blood cells are going to take on a sickle shape, which means it cannot latch on to oxygens properly. Therefore, that person will be oxygen deficient. So the red blood cells of these individuals contain a specific hemoglobin that causes it to have this shape. And so that's going to be hemoglobin S or HbS. And that causes that red blood cell to bend into a curved shape or a sickle shape. And that in turn causes it not to be able to carry the oxygen properly and to take it and be able to give it up to that interstitial fluid where it's needed. Now looking here, you can see a red blood cell, and at the bottom you're going to see a sickle cell anemia red blood cell. In order to have sickle cell anemia, it's only going to be one amino acid difference or one amino acid off that changes in the sequence that causes the sickle cell shape. And of course, this is genetic. Now, people who are going to be carriers, this means that they have one good gene for red blood cells and that they have one that has a sickle cell anemia. So they inherited either from their mother or their father the gene for sickle cell, but then they inherited a normal red blood cell gene from one of the parents. So that would be making them a carrier. With these people, they are not going to have the full symptoms of having sickle cell anemia, but it does make them immune to malaria. They do not get malaria. Now, if both parents gave the child the sickle cell gene, then that person would be considered to have sickle cell anemia. You're going to see sickle cell anemia prominent in areas that are going to be close to the equator. And that's because lots of times it's kind of a genetic mutation that did help in those areas because they have lots of mosquitoes, and it would actually help them if they were carriers to not be susceptible to malaria when they were bitten by the mosquito. Okay. Platelets are often referred to as thrombocytes. These are going to be fragments of a large cell that's called a megakaryocyte. These, the hormone called thrombopoietin is going to stimulate the production of these megakaryocytes. And then once they start to break down and become mature, that's when they become the full platelet or thrombocyte. These are going to be important because they're going to help seal off bleeding of vessels within the body, or if you have some type of cut or opening to the outside portion of the body. Now, a normal thrombocyte count is going to be about 150,000 to 400,000 per microliter of blood. If you're looking here at this picture, the little pieces that you see that kind of have a pinkish purplish tint to them, those are going to be the platelets. So that's what you're looking for if you're looking at platelets. Now, when you're forming a platelet, we start out with the hemocytoblast, which is going to be the stem cell. It will then develop into that megakaryoblast, and that's going to be a stage 1 megakaryocyte that you see there. Notice how it has a nice nucleus and it's nice and clear in the cytoplasm. Once it goes into stage 2 and stage 3, that megakaryocyte then starts to become granulated looking. You start to see the nucleus start to take a different shape. Then going into stage 4, that megakaryocyte actually starts to break down into pieces. And so once it breaks down into pieces, then it becomes mature platelets. All right, blood plasma is going to be 92% water. Remember, about 55% of a blood sample will be plasma. So 92% of that plasma or that liquid is going to be water. The protein portion is going to be made up of different proteins called albumins, globulins, and fibrinogen. Now, albumins is going to make up 54% of the proteins that you'll find in this plasma. These are going to be made in the liver, and they're going to function to create osmotic pressure. So water tends to want to stay in capillaries, so it's actually going to help to bring the fluids from the capillaries out and into the different tissues. And so that will help things come across, and it'll also be able to bring it from the tissues back in. And so edema is going to result when there's not enough albumin proteins that are going to be found in the blood, so it's not able to take the liquid from the tissues and put it back into the bloodstream. The next protein is going to be globulins. This is going to be about 36% of the blood proteins that you'll find in a blood sample. You have both alpha and beta globulins that are going to be made by the liver and function to transport lipid and fat-soluble vitamins. Now with the gamma globulins, these are going to be the antibodies. They're made by the lymphocytes and they help to function and fight disease. Like we said with the alpha globulins, these are going to be the ones that help transport lipids and fat-soluble vitamins. Those fat-soluble vitamins are going to be vitamins that once you take them, they will be stored in the adipose tissue or the fat, and as your body needs them, it can extract them and carry them to places that need them. Now, if you take too many, they can become toxic or be at toxic levels. Water-soluble vitamins, things such as vitamin C, for example, if you take too many of those, your body simply flushes them out with the urine, and you don't overdose on those basically, as long as you don't take extreme exorbitant amounts. So the fat-soluble vitamins, things like vitamin A or vitamin E, those are going to be the things that will be stored in the tissues or the adipose tissue, and they are carried around in the bloodstream using these, the alpha globulins. All right, next is fibrinogen. This is going to be 7% of blood proteins. This is going to be made in the liver, and it's involved in forming blood clots. So fibrinogen is then going to help create fibrin, and that's going to create almost like a net-like form for platelets and blood cells to stick to in order to stop hemorrhaging or stop bleeding. All right. Plasma gases, including things like oxygen, are going to be important in helping cells metabolize nutrients. Carbon dioxide is going to be cellular waste products. And then you have nitrogen, which is going to be taken in when you breathe in and exhaled when you breathe out. And that simply has no function that we know of. The nitrogen that your body uses will be taken in through food products. Plasma nutrients are going to include things such as amino acids, simple sugars, nucleotides, and lipids. With these nutrients, the amino acids, of course, are going to come from things such as food, proteins that you eat, so it could be different types of meats or beans. The simple sugars are going to be the carbohydrates that you're taking in. The nucleotides are simply going to be from the nuclei that are going to be found in the cells of these foods that you're taking in. And then, of course, the lipids will be the fats that you have in these foods. Non-protein nitrogenous substances are going to include things such as urea or uric acid that can be found in the bloodstream. These are going to be formed from the breakdown of amino acids and nucleic acids and are going to be excreted in the urine. And also some are going to be excreted lightly throughout the skin or through the pores of the skin. You can excrete some of that urea. Plasma electrolytes are going to include things such as sodium, potassium, calcium, magnesium, chloride, bicarbonates, and phosphates. These are going to help maintain things such as pH, and of course you'll have these sulfate ions. Now here's a video that you can watch on your own time if you want to in the PowerPoint. It just simply goes over plasma donation, and actually you can make a little bit of money by donating plasma. I know there is a plasma donation center in Danville, and I do work in Danville during the school year part of the time, and so I know a lot of people who actually donate plasma, and it's a lot of extra money that they get from it. So, and especially if they encourage others or sign others up under them, they make an extra bonus doing that. So if you need a little extra money sometimes, this will greatly help others because they'll be able to use the different proteins and different things. They say up to 7 people can be helped by 1 plasma donation. And so by doing this, you're helping others and you can make a little bit of money. Hemostasis is going to be the ability for the body to stop bleeding, and it's going to be accomplished in 3 ways. First thing you're going to have is going to be what's referred to as a blood vessel spasm. This is going to be the contraction of the smooth muscle that's surrounding the blood vessel. It's going to help to lessen or stop the bleeding for a short period of time. Next, you're going to have a platelet plug come in while it's spasming, so it's trying to stop that blood. The platelet plug will come in and form. So the adhesion of platelets to this damaged area of blood vessel is going to come in, and it's going to help stop or lessen the bleeding. And it actually works best, of course, on small breaks. And then, of course, the third thing that's going to happen is going to be coagulation. This is where, remember, you had your fibrinogen, creates that fibrin, and that forms a mesh network where those red blood cells and platelets are going to come together and stick to it and form a blood clot to stop that bleeding or stop leaking of blood through that area. And so here's simply a diagram going over showing you what's happening. You have first the tissue damage, so you have the blood spasm, platelet plug formation, and clotting mechanism. There you have that prothrombin activator. It activates that prothrombin and comes together with the calcium and creates thrombin. Thrombin then goes in and you have fibrinogen. It adds in more with more calcium and it creates fibrin. And then the blood clot is actually going to be coming together in formation at that point. And so looking here at this picture, you see the red blood cells, you see some chunks in there, which would be the platelets, and of course you see lots and lots of that fibrin or that mesh network that was created for all of these things to stick to. Put it there for now. So once a clot forms, it is going to then consolidate. So then it starts to tighten and draw the edges in of that damaged blood vessel. From there, it's going to use vitamin K for that normal clot. So although it's not directly involved, it does actually need it in order to make the substances for those clotting factors in order to create that clot. And then small unwanted clots sometimes could happen, but they're usually dissolved by plasmin within the blood, and that's called fibrinolysin when it's able to do that. And so every now and then you might get these small blood clots, especially if you're traveling in an airplane or you've been sitting for long periods of time. Okay. So an abnormal blood clot formation on the wall of a vessel is going to be referred to as a thrombus. So over to the left-hand side, you see what a normal artery wall looks like. If you look over to the right-hand side, this is going to be one that is forming a clot and that's starting to be blocked. So if the thrombus then breaks away or breaks loose from the wall of that artery, it's then going to be referred to as an embolus. Now going into blood, with blood we're going to have what we refer to as blood groups. So we have the ABO blood groups, and this is simply going to be what type of proteins are going to be found on the cell membrane of that red blood cell. So if you have type A blood and you look over to this picture that's going to be in the top left-hand side, you're going to see that that red blood cell has a specific shape of protein that's going to be found on that cell membrane. That specific shape we're going to call A. Now, it is going to clot or coagulate together and clump together if it's exposed to any type of anti-B antibodies. And so with blood type A, it's going to make anti-B antibodies. And so if any type of B blood comes in, it will clot with that. Looking over to the right-hand side on the top, you see type B blood. Now notice that's going to be a different shape. People with type B blood will have antibody anti-A. And so with that one, anytime it is exposed to type A blood, it will cause coagulation or clumping together of the blood. Now this could be fatal. With type AB blood, it doesn't make either antibody A or antibody B for there. So you're not going to have either one of those anti-A or anti-B. So you can take in type A blood, type B blood, or AB blood. With the type A blood, you can only take in type A blood or type O blood. With type B blood, you can only have type B blood or type O. Now looking down at the bottom, we see that we have type O blood. Notice how, with this one, there's not going to be any type of proteins that are going to be found on that outside cell membrane of the type A or the type B. And so this is referred to as type O. Now people with type O blood will have antibody anti-A, and they'll have the antibody anti-B. And so that means that if they're exposed or have B blood, AB blood, or A blood put into the system, then it will cause a clotting or coagulation, and it's going to clump up, and it's not going to flow well, and it could result in death. So looking here at this chart, we see how the different blood types, which ones they agglutinate or which ones they have antigens for, and what their donors could be and what their receivers can be. So with type A blood, we have the agglutinogen or antigens for this type. And so it has an anti-B, so it will coagulate or agglutinate with the B blood if B blood comes in. It is compatible with A and O, so that means it can take blood in from type A or type O. It's incompatible with type B or AB. Type B blood has the anti-A, and of course it can receive from type B or type O, and it cannot receive from type A or type AB. AB blood has both A and B antigens on the outside, and so the antigens are those proteins that you saw on the outside of the cell membrane. And so it does have the A antigen and the B antigen. It doesn't have antibodies for either A or anti-B. And it can receive from A, B, AB, or O. And there's nobody that it's incompatible with as far as receiving. For O, it has neither antigen. It does not have antigen A or antigen B. It has anti-A and anti-B for its antibodies. It can only receive blood from O, and it's incompatible with A, B, and AB. And so here that you see at the top, these are going to be antiserums. Untreated blood is going to be what's on the left-hand side. And so if you were to use anti-A and anti-B with the first blood type, you see that it's going to be type A. Now with this type A, we know that it's type A because it clumped or it coagulated with it. So that tells us it's type A. With the second sample, we know it's type B because we see coagulation or clumping with B. The third sample we know is AB because we see the clumping and coagulation with both. And with the last sample, we know it is type O because it is smooth throughout. So it's not going to react with those antiserums. Now, lots of people are probably thinking, what happens to the positive factor? That's not included with this type. That's simply going to be another antigen that you're going to find on the outside of the red blood cell's cell membrane. But that's not shown here. So looking at popular groups and blood types that you will find normally in these different groups. You can go through and look at these different types. Actually, the most popular type of blood is going to be O positive, and the rarest type of blood is going to be AB negative. Now, Rh blood groups, that is what I was talking about with the positive or the negative. And it gets its name Rh from the animal that it was first found in a rhesus monkey. So rhesus monkey blood is the first blood that they actually found this protein in. And so that's where it gets its name. So for example, I am O positive, and so that means that I have no A or B antigens on the outside of my cell membrane, but I do have the Rh antigen or protein on the outside of my cell membrane. And so I am referred to as an Rh positive blood group. Now with a mother who is Rh negative, lots of times you know that they are incompatible when they have a fetus that has positive blood. So usually it doesn't occur until after the first pregnancy. Usually the first pregnancy goes smoothly, goes okay. But during that pregnancy, the body will create those Rh antigens. And so it wants to fight off any subsequent pregnancy after that with a embryo that's going to be Rh positive. And so now that person has anti-Rh antibodies in them. And so from that second pregnancy on, it will actually attack that fetus as it's starting to develop and grow. So what do we do about this? Actually, with this, there is a shot that they can take called a RhoGAM shot. And this shot will suppress the mother's immune system so it cannot attack the fetus. And therefore, the fetus is allowed to grow and develop, and it's born perfectly normal and healthy. Okay. Stem cell transplants from bone marrow and cord blood. With the bone marrow transplants, these are going to be performed to replace cancerous red bone marrow with normal red bone marrow. The donor's bone marrow is going to be usually collected from the iliac crest of the hip bone. Stem cells collected from the umbilical cord can also be used. So lots of people after birth like to take that umbilical cord and they will freeze it and they will keep it in a cord bank. It can also be used or have advantages over bone marrow and bone transplants because it's coming directly from that person. And if other siblings are close in genetics with the cord that you saved, then it could possibly be used for them as well. And so it is a specific fee. You do have to let them know before the baby's born. That way they have everything ready to go once the baby's born to take that cord blood and send it off properly and store it properly to the cord bank, which then will use a cryogenic lab, which will then freeze it and save it in case it's needed in the future. And it is, I'm sure, there's an initial fee, but then I think there's also a monthly fee that you might pay with that. And here is a simple video on hepatitis C. Hepatitis C is going to be an important disease that attacks the liver, and it can't be prevented as of now with a vaccine. Last time I checked, they did have some medications that were out, but we have certain vaccines for some hepatitis, but right now they're working on this one, I'm sure. All right, and that's going to end our video on blood, and I will see you with our next unit.